In this video, we'll be discussing about multiple alleles. First of all, if you want to know what allele is, you can watch that video also by clicking the link above that slides in here. Now getting back to the multiple alleles, we see when there are more than 2 alleles for a gene within the population at specific loci. At that time, we say within the population the multiple alleles occur. Do not get confused here that a gene is having multiple copies of allele in a single individual, but the multiple alleles occur within the population. So in that way, genotype has only 2 alleles for a gene. But when we talk about multiple alleles or multiple allelism, it's a gene controlling any phenotype which is having different copies of alleles found within the population, that too on a specific loci. The first property of multiple alleles is that they show codominance. It's when the heterozygous condition occurs in the individual, both the alleles express themselves without repressing each other. In the human population, we see we have got a best example for multiple alleles in the form of ABO blood group system. In the ABO system, the chromosome number 9 has got ABO gene on its long arm Q as shown in the figure. For this ABO gene, which is usually denoted by I letter in the books, it has got different versions, what we called as alleles. There are 3 alleles for that. We have A allele, B allele and O allele. Only one among these 3 alleles will be present on one chromosome at a time. Then we will get one allele from mother, one allele from father, and in an individual we will get 2 alleles. So what are these alleles coding for? The A allele codes for an enzyme that adds galactosamine on surface of RBCs, whereas the B allele codes for an enzyme that adds galactose on surface of RBCs. And finally, we have O allele. It does not add any sugar residues towards the surface of RBCs. Basically, the O allele is a null allele. The protein that we get from this is non-functional, whereas both A and B allele are able to produce transferase enzymes, which we are going to see what these transferase enzymes do in the next video, where we will be discussing about the biochemistry of ABO blood typing system. As you can see in this picture, it also illustrates the chromosome number 9 having 3 different alleles at the position of Q34.2 position of Q arm. There are 3 different alleles for a single gene, but this occurs within the population, not in an individual. In an individual, only 2 alleles can occur. As we have already seen, the gene that encodes for enzymes that add sugar molecules on surface of RBCs is ABO, and it is designated as letter I. And this I gene has 3 alleles, as we have already discussed it, which can be represented as IA, IB and I0 or IO. When there is IA, the RBC has A antigen. IB gives B antigen to RBC, whereas the I0 does not add any surface antigen to RBCs. But it must be noted that when the O allele does not go for any addition of molecules on RBC surface, that time the RBC is left with precursor molecule that's already present on its surface, what we called as H antigen. This precursor molecule, what we called as H protein or H antigen, is also present in A and B type individuals because this is the precursor molecule. Now let's see the cross between male and female with possible alleles after segregation. For this cross, female will give half of the alleles and half of the alleles will be given by male. From females we will get IA, IB and I0. And we also get the same set of copies from male too, that's IA, IB and I0. From this cross we get IAIA, IAIB, IAI0 and so on. In this cross we find out that 3 homozygous condition occurs that are found within the cross in the form of IAIA, IBIB and I0 individual. Rest are all heterozygotes. When we see this cross, we get 6 possible conditions as IAIA condition. It's the individual having A blood group. Then we have IBIB condition. It is the individual having B blood group. Then we have IAIB condition, individual having AB group. Then we have IAI0, individual with A blood group. Then we have IBI0, individual with B blood group. Then we have I0 individual having O blood group. If we analyze these combinations, we get 4 types of blood groups that are found within the human population as A type, B type, AB type, and O type. So this is how the multiple alleles or multiple allelism occurs within the population. So this is all about multiple alleles with the explanation from ABO type system. In the next video, we'll be discussing about the biochemistry of ABO type system. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.